Hello again. In this video, we'll take a look at cell structures. We're focusing in this video on the endoplasmic reticulum. Now remember, when we're thinking about cell structures, we want to think about how they're working together. So if you've got a cell, an eukaryotic cell with its nucleus, the nucleus contains all of that genetic material. Pieces of the genetic material, pieces of DNA called genes, are copied into RNA molecules right here in the nucleus. Those RNA molecules are shipped out of the cell, out of the cell's nucleus and into the cytosol. The RNA molecules are then used by ribosomes. Ribosomes attach themselves to them and start making proteins. Okay? There are lots of other structures inside eukaryotic cells. And so we want to think about what they're doing. So we're right here looking at cell structures. Have to get there. Okay. Many of the membrane-bound organelles are a part of the endomembrane system. We consider the endomembrane system a system of internal membranes that are found in eukaryotic cells that help to divide the cell into structural and functional components. They're a system of organizing, right? The membrane system basically organizes different parts of the cell, kind of contains them, keeps them organized, helps them do their job, so that they can function as a part of the bigger cell. The endomembrane system includes all sorts of membrane-bound structures. It even includes the plasma membrane and the nuclear envelopes. It includes the endoplasmic reticulum. It includes the Golgi apparatus. It includes lysosomes. We'll talk about those. It includes vacuoles. And maybe a few other things. But the function of the endomembrane system is basically to synthesize molecules and transport molecules to their appropriate location. To synthesize molecules like proteins and lipids and transport them to their appropriate locations. The endoplasmic reticulum is one part of this network of membranes, internal membranes. It is typically attached to or very close to the nuclear envelope. And it's a system, it's a little net of membranes, essentially, a little set of tubes, membrane-bound tubes. And sometimes I draw it something like this, membrane-bound tubes that kind of are a big component of each cell. Each membrane-bound tube has a membrane on the outside, right? That would be on the edge. And then the space in the middle is sometimes called the cisterna or the lumen. So if you hear these terms, like the cisternae would be the material or the space inside the membranes. And this is a huge component of the membranes found in the cell. So what's happening at the endoplasmic reticulum? Well, one thing we want to remember is the inside of the endoplasmic reticulum is filled with all sorts of enzymes, and even the membranes of the endoplasmic reticulum have enzymes in them. So what does this system of tubes do for the cell? Well, there are at least two different kinds of endoplasmic reticulum. Some of the endoplasmic reticulum we call the SER, or the smooth endoplasmic reticulum. The SER doesn't have ribosomes attached to it. The RER, the rough endoplasmic reticulum, has ribosomes attached to it. And remember what ribosomes do. Ribosomes are the site where proteins are synthesized. So The rough endoplasmic reticulum is probably doing things with proteins. The smooth endoplasmic reticulum may have slightly different functions, and in fact they do. So let's take a look at some of the functions of the smooth and the rough ER. And I wrote some of these down. They're not in your notes, so let's think about them a minute. They're very different. They have different jobs to do, and they have a different structure, right? So the SER has enzymes that helps the SER to synthesize all sorts of lipids. Things like fatty acids and phospholipids that become parts of the membrane, and steroids. So SER in cells in, say, the testes are doing the job of making testosterone, for example. Another function of the SER is to detoxify drugs and poisons. The SER has enzymes that enable this part of the cell to take poisons and make them less poisonous, to make them more water soluble so they're easier to get rid of. And so when we think about detoxification inside our bodies, we think about the liver. The liver, that's where our bodies do the job of detoxifying poisons that we come into contact with. And in fact, that's true. And guess what? The cells in the liver have an abundance of SER, and those SER structures help to detoxify the poisons that the liver cells come in contact with. Another function of the SER is in muscle cells. In muscle cells, there's a specialized kind of SER called sarcoplasmic reticulum. And that sarcoplasmic reticulum does the job of like sequestering or holding in calcium ions and releasing them. And that helps muscle cells to contract and to relax. Another thing that the SER does is it plays a role in carbohydrate metabolism. So when we've got something like a big glycogen molecule, it can break it into smaller glucose molecules. There are enzymes here in the SER that do it. Or it can build new glycogen. The RER, remember, is that network of tubes or flattened sacs that has ribosomes attached to it. And ribosomes are the site of protein synthesis. Once these proteins are made, so if you've got a ribosome sitting at the surface of the rough ER, the protein that is generated, that's just that chain of amino acids, if you will, is shipped into the inside of the rough ER. And the rough ER then is involved in dealing with this protein. It's involved in the production of protein because the ribosomes at the surface are making it. But then the string of amino acids needs to be modified. Sometimes pieces have to be added to it or cut from it. Little sugars have to be added or removed. That happens inside the rough endoplasmic reticulum. Sometimes the amino acid chain has to be folded, right, into its three-dimensional shape. And that folding takes place in the rough endoplasmic reticulum. There are even special molecules called chaperones that help to fold up our proteins into their three-dimensional shapes. Sometimes there's quality control that takes place. So if a protein is being made and you know, maybe it doesn't have quite the right shape, it took on the wrong shape, there are enzymes that recognize that, oops, this is a mistake. This protein was made incorrectly, and so they will destroy misshapen proteins right here in the RER. And then eventually, when the proteins are modified and checked for quality, they leave the rough ER in what are called vesicles. And these little proteins then leave the rough ER in small vesicles. These modified proteins will head in the vesicles toward the Golgi apparatus. Sometimes they head toward membranes, like toward the plasma membrane, let's say, of the cell. But then they're shipped toward their next phase of development, because these proteins are typically not not quite mature yet. These proteins aren't quite ready to do their job, but chances are they're going to go through the Golgi apparatus and then get shipped to their proper location. Second response question of this chapter says, what is the function of liver cell smooth ER in the metabolism of drugs and alcohol, and how might the liver cells of an alcoholic differ from those of a non-drinker with respect to SER in each cell? This question doesn't say anything about cirrhosis of the liver or, cells dying. It asks about the smooth endoplasmic reticulum. This is an extra credit question on our extra credit assignment, so check it out. And that's it for the ER.